think a lot of really important things came forward, and I'm hoping that's going to stimulate some questions from you guys. So I'd like to open the floor to questions. I see one over there.
So about 25 years ago, I did a radio talk show for Wisconsin. It was a monthly 1-hour call-in radio talk show about science. We talked about volcanoes. We talked about dinosaurs. We talked about, I don't know, exploding stars. And in the 9th month that I was doing this, they said, let's talk about asbestos. And boy, did I get in over my head. Because I made a comment that there are different kinds of asbestos-formed minerals, and some of them are extremely dangerous for anybody, but others, like the chrysotile, becomes much more dangerous if you're a smoker. Well, boy, oh boy, I got fired. Right, I wasn't getting paid, but I got fired. What I want to know is, was I wrong? And this must be like it must have been a class action lawsuit, and a bunch of people who were smokers were hoping to get a big payout out of it. I think that's what happened, although there certainly was a lot of problem. What's this deal with smoking and asbestos?
So the early epidemiological literature on asbestos workers showed that those who did not smoke did not develop lung cancer, even though they had high exposures to chrysotile. At a rate that was, well, maybe a tiny bit elevated for the worst exposures. So I think that under the highest occupational exposures that existed, you could develop lung cancer and not smoke. But the majority of the lung cancers are attributable to the combination of the two, and they're synergistic, increasing the potency by 100 times. So if you worked in asbestos and you smoked, you're going to get lung cancer.
I think one of the biggest problems is that a lot of us don't want to talk about these issues. Your example is exactly right. Whenever we do these things, we tend to get in trouble. As Gordon said earlier today, we tend to open the can of worms. But I think a lot of us need to be doing a lot more of this. And as Matt showed, when you can't tell an anthophyllite from talc, that's kind of a problem. And this is happening all the time.
I would like to just draw away a little bit from asbestos, because there are a huge amount of other minerals in the air that are maybe equally dangerous, maybe much more, just because of the number of particles we put out there. There are 4 to 5 million people that die prematurely every year from air pollution. That's more than twice when we compare to water pollution. And these particles in the air are so poorly characterized. And so they are also mixtures. It's not just one mineral, but it's many minerals. They have adsorbed species on there, for example, polyaromatic hydrocarbons. So we have to look at the surfaces of these particles as well. And that's a hugely important thing also in the asbestos toxicity, because the surfaces are ultimately the things that are in direct contact with the body fluids. They can create reactive oxygen species that trigger all these other effects in the body. So this is an incredibly interesting but highly complex topic. And it's not just asbestos. We put out tons of diesel soot, which is a carcinogen, much more abundant than asbestos in the air. We put out tons of magnetite, which we all have in the brains for various purposes, but we also accumulate magnetite in our brains just through inhalation of magnetite that is put out by combustion processes. So mineralogists have a lot to do there, and it's a highly exciting field with lots of unknowns.
Yeah, I'd just like to add one thing. I know in a lot of the, I think it's going into the medical field, a lot of them are very centric, like it's just, like the focus is so narrow into asbestos for a lot of them. We don't know just generally what are people breathing in. Nobody's measuring total particulate, what's all in people's lungs that have disease. They're only looking for known causes of the disease from like a pathology perspective. And it's usually pathologists doing that work, not mineralogists to characterize those materials. So,
I'd also like to say that the biological models for understanding variability of mineralogical properties are very rudimentary and are not robust at all. So the first introduction that talc caused ovarian cancer was done on a bunch of rats, and they gave, I think it was instillation, or I've forgotten how they got the stuff there. Probably. And anyway, so the rats developed tumors, but the problem is that you couldn't give those rats any particulate at all that didn't have tumors as a response. There was no differentiation among the minerals. And so I see these people do a lot of cell work. There are some enlightened investigators, but by and large, they don't have a control with the non-fibrous variety of the same mineral or anything like that. It never occurs to them. So we don't really know what characteristic is the response that they're getting associated with.
Well, I just wondered, my takeaway from part of this talk is that MSA perhaps should lobby for a certification program for these mineral identification labs.
There is a national certification program.
Well, I mean, it sounds like it's not. It sounds like it's not working.
No.
Well,
if people
the
can't
reason
demonstrate they can't separate talc from coming tonight or something like that.
Well, the reason it doesn't work is because the certification program is for a laboratory, but also the only samples ever sent to the labs will be amosite, chrysotolite, or chrysotile. You know what I mean?
Well,
They're not testing the labs to these other things.
You could have a wider variety of samples that are part of the validation process. I think that should be something. That's a position paper of the society to do, is to have rigorous certification for these labs that are involved in these testing.
I don't disagree with that.
I think that would be fantastic. I mean, it would be an additional certification outside this national certification program, which is worthless.
And they could probably get paid better then, and you'd get better quality.
Yeah, and they could brag about their ability to actually do the job.
Any more questions out there? I think we can do one more. No, we'll do two more.
Okay.
We'll start over here and then to you.
That's all right. Big.
Here we go. All right, thanks. No, I appreciate the talks, and you brought up at the end sort of the uncertainty that is in the medical field about what the toxicological mechanism for these diseases from these minerals are. And I guess my question is about where do we go from here as a group of mineralogists to understand what the fundamental aspects of these minerals really are that may be causing these diseases in terms of their physical properties, their chemical properties, their surface properties, their size properties, and how do we sort of drive this forward to better constrain the toxicology and the dose-response relationships that are really at the root of what should be defined for defining a toxic response.
The problem that we have is we don't have anything to correlate against. And the biological models, the cell studies and that sort of thing, some of those are pretty good and maybe that they're useful. And so that's the first part, is you need you've got the minerals, you can vary characteristics or study them, but they have to be be responded to by a biological system in some variable way so that we can correlate. These are the data that I showed you are the only data that I know that actually look at variability of the potential to cause mesothelioma, and they were just published. So we just have a big problem in quantifying effects of minerals in the kind of way we think of things. You know, two variables, and we're going to independent, dependent, and we're going to, the way we think, and it just is very, very difficult in this particular system.
Yeah, I think the other thing where to go though is, we've been, I don't know who we are, but generally been stuck in this paradigm of this 3-to-1 being important, and it's never been correlated to be important to anything. But that's where we're just like stuck with this 3-to-1. We can't move on from a 3-to-1 aspect ratio. There were some proposals, I think it was in 2003, they were looking to update the risk models for the EPA IRIS risk assessment stuff. And they were proposing, what was it, 10 microns and longer and 0.4 micron widths or something like that. And there was a lot of promise, all the comments in the meeting seemed really good, but it didn't go anywhere. It's been 16 years later and it's just, we haven't progressed. We is a much larger group than me, but.
We know the dimensions are a primary variable. There's no question about that. I mean, the width controls access to your lungs. It controls translocation within your body. It is a primary variable. It's the other ones that we don't have any sense of. What is aragonite? What about the fibrous clays? What about fibrous talc? What about other fibers? How do they respond? And how does the biological system respond, where dimensions are not an issue?
So. One more question.
Okay, it's in the form of a comment, though, and an encouragement. As mineralogists, we claim our minerals, but too often material scientists and other scientists claim our materials as theirs. I think it's time for mineralogists to take back synthetic nanoparticles that we are uniquely situated and well trained to actually characterize these well, I think better than material scientists. I have a foot in both fields, and I think the geology counterpart does a very good job of properly characterizing things when you're trained properly. But things like magnetite and manganese and welding fume and all of these other particulate matters are just ripe for the mineralogist to take back.
All right. I'd like to thank the speakers again.